Okay, so we're going to talk about synaptic clefts now and kind of what happens at synapses. And this, of course, is when the neurons need to communicate to one another. Because remember, all of these dendrites that we see on the neurons, in this case, it's a myelinated neuron, all of these dendrites have the capability to communicate with other neurons. And so here you can see that there's two neurons kind of connected together. And so we're going to talk about how this actually happens in terms of how do we get that impulse from the originating neuron, known as the presynaptic neuron, to the receiving neuron, which is known as the postsynaptic neuron. So just like in our descriptions here, the synapse basically is the gap between the communicating neurons. So if you take a look here, we have our axon, and we have an impulse traveling along this axon. You can see it says signal direction here. And then basically what happens is you have this little space where the presynaptic neuron, the one that is carrying the impulse towards another neuron, and the space between that and the postsynaptic neuron is called the synapse. So if we take a look at this a little bit closer, again, we can see our kind of classic myelinated neuron here. We have our dendrites, we have our cell body, there's our nucleus, and of course our transmission of our impulse is going to travel along the axon, right, all the way to the axon terminals. And then down here, eventually, we can see that one of those axons actually comes into contact with the dendrite of another neuron. So that's how they communicate to one another. And so if you kind of take a look at that little square and we kind of blow that up here, this is what we actually see. So you can see the synapse then, by definition, is the place at which the axon of the presynaptic neuron is meeting up with the dendrite of the postsynaptic neuron. And then kind of what happens there? How does that impulse actually get transmitted? So once the impulse has traveled through the entire length of the axon of the presynaptic neuron, it's going to arrive at that axon terminal, which again is that connector point between the presynaptic and postsynaptic neuron, and then that space between, okay, so you can see that the presynaptic neuron axon and the postsynaptic neuron dendrite don't actually touch. You can see there's actually a little space there. Now this whole thing is called the synapse, but that space in between is called the synaptic cleft. And one of the things that I want you to pay attention to is that those two parts of those nerve cells don't actually touch each other. You have that space, you have that synaptic cleft. And so the impulse, the electrical impulse, doesn't really have the means by which to actually jump over that synaptic cleft. And so we have to have a mechanism by which to get that impulse across. And that's where chemicals called neurotransmitters come to play. So if we take a look at our diagram here, once again, you can see that we have our presynaptic neuron here. This is our cell body, right, of our nerve cell. This is our nucleus here. And then, of course, that electrical impulse is going to be traveling along the axon of that presynaptic neuron. And eventually, it's going to find a dendrite of a postsynaptic neuron. And then that space at which that axon on the presynaptic neuron and the dendrite of the postsynaptic neuron communicate with one another, that is called the synapse. The space between called the synaptic cleft. So the axon terminal then, okay, contains numerous what we call synaptic vesicles. And if you remember from, I don't know, Science 10, Biology 20, synaptic vesicles are like little bubbles, for lack of a better word, little bubbles that actually contain neurotransmitters, okay? So vesicles are little transport structures within cells that allow materials to be moved around a cell, moved out of a cell, moved into a cell through endocytosis, exocytosis. So when we talk about synaptic vesicles specifically, these are the vesicles that contain neurotransmitters, and they of course are found in the nerve cells. We also have a lot of mitochondria there, and remember from our Bio 20 studies as well, mitochondria are where the cellular respiration takes place for the production of ATP, because of course ATP is the energy molecule that all cells need for all metabolic processes. So the whole purpose of those little vesicles, those little bubbles, is to actually contain neurotransmitters that allow for the transmission of the impulse from the presynaptic to the postsynaptic neuron, so it can continue all the way until it eventually gets to the effector, which is that gland or muscle or whatever the target the target of the impulse. So neurotransmitters will move across the synapse, carrying the impulse from one neuron to another, and that's because that synaptic cleft, or that space between, is actually too large for the impulse to actually leap or jump across. So the neurotransmitters are used to actually facilitate the transmission of that electrical impulse from the presynaptic to the postsynaptic neuron. So presynaptic and postsynaptic terminology that we have to be very comfortable with. Okay, so when we get into the sequence of synaptic events, I know it'll seem a little bit confusing at first, but it's actually not that bad. Let's just orient ourselves to our diagram here first. So this here, of course, is going to be our presynaptic neuron. This down here is going to be our postsynaptic neuron. Okay, so pre, that's where it originates. Post, that's where the impulse ends up. And then, of course, the space in between the postsynaptic and presynaptic is called the synaptic cleft. The whole thing collectively together is called a synapse. Okay, so the presynaptic, the postsynaptic, the cleft all together, that's called the synapse. Okay, but the space specifically on its own is called the synaptic cleft. Okay, once again, you can see that the space between the presynaptic and postsynaptic neurons is relatively large, and it's too large for the impulse to actually move. So that's where our neurotransmitters come into play in terms of facilitating the movement of that electrical impulse through that synaptic cleft, through that space, and to the receiving postsynaptic neuron. So when we say the term action potential, just remember that action potential is kind of another word for that impulse, okay, or that electrical impulse. So we have that electrical impulse, and you can see it here, these green kind of arrows that I just kind of covered over. We have this electrical impulse traveling down that presynaptic neuron, so it's gone all the way through the axon, that long axon, and eventually reaches the end of the axon terminal, and then it's kind of like, how do I get from that presynaptic to that postsynaptic neuron? And that energy, again, is called the action potential. Now, what basically happens is when that action potential makes its way down towards that synaptic cleft, what that does is it facilitates the fusion of the synaptic vesicles, okay, these little guys here that contain those little purple neurotransmitters. It actually pushes them down towards the membrane here, and it causes those little vesicles to fuse with the presynaptic membrane rather. And then notice what happens here. All those little neurotransmitters are dumped into that synaptic cleft, okay? And remember the process by which we have vesicles fusing and then releasing materials, that's called exocytosis, right? So those neurotransmitters, once those vesicles fuse with that membrane, those neurotransmitters are going to be released by the process of exocytosis into that synaptic cleft. And basically then what they do is they move or they diffuse across that synaptic cleft towards the postsynaptic neuron. So the neurotransmitters will then find very specific little receptor sites on the membrane of the postsynaptic neuron, and this triggers sodium channels to open. So what I want you to notice here is you have these little guys here, okay, these little kind of pinky purpley guys here, and you'll notice that the shape of them is specifically designed to receive that little triangular shaped neurotransmitter, okay? So as those neurotransmitters are diffuse into that synaptic cleft and then they diffuse towards that postsynaptic neuron, those little neurotransmitters are going to bind to these receptor sites on the postsynaptic neuron. So that, remember, the postsynaptic neuron, that is the dendrite of the receiving nerve cell. And then notice what happens here. Once you get those little neurotransmitters binding with those receptor sites, that causes sodium channels to actually open up so that sodium starts to flow into the postsynaptic neuron. Okay, so that's very significant. So the neurotransmitters bind to those receptors, that triggers the opening of those sodium channels, and then sodium starts to flow into that postsynaptic neuron. Now, this process of the sodium flowing in is called depolarization. And I want you to remember that sodium, basic chemistry thing, is a positive ion, right? So positively charged. And this actually causes the postsynaptic neuron to become more positive as that sodium is allowed to flow in to that neuron. Once we reach a certain threshold of positivity, okay, in that postsynaptic neuron, we have reached what's called an action potential. And then that's when the impulse is actually then allowed to move and continue to flow down the axon, move from down the axon of the postsynaptic neuron, okay? So once that happens, once we have those sodium channels opening, sodium flowing into that postsynaptic neuron, and we get more and more and more and more positive charges building up in there, then the nerve impulse is going to travel down that axon in that postsynaptic neuron, and then the same process will happen again as it communicates to another neuron, another postsynaptic neuron. So there's two types of neurotransmitters, okay? So we talked a little bit about neurotransmitters and the fact that they allow the transmission of the impulse from the presynaptic to the postsynaptic neuron, right? So we have excitatory transmitters, and those are definitely the most common types of transmitters. And remember what excitatory ones do. So this is like the example we looked at the previous slide. Excitatory neurotransmitters cause those sodium channels to open so that sodium can flow into the postsynaptic neuron. This is called depolarization, okay? So anytime you're getting more positive charges building up in that postsynaptic neuron, that brings us closer and closer and closer to something called threshold potential. And we'll talk a little more about threshold potential in the next presentation, but threshold potential basically is the point at which the neuron can actually fire and move that electrical impulse along the axon to a new neuron, okay? So these are called excitatory neurotransmitters. It's kind of common sense because they depolarize the neurons by allowing sodium into the cell, and they allow the cell to reach that threshold potential, which is the point at which it can actually fire that impulse down its axon and eventually communicate with the new neuron or an effector or the muscle or the gland if it's at the end of the road for that transmission. Now, inhibitory do the opposite, okay? So the names kind of make sense. Excitatory, the nerve cell is going to be excited and I can move that impulse along. Inhibitory, it's like, oh, it's inhibiting, it slows things down, it doesn't allow transmission of the electrical impulse. So instead of triggering sodium ions and sodium channels to open and allow those ions to flow, inhibitory neurotransmitters actually trigger chloride channels to open instead of sodium. And remember that when we talk about chloride ions, they are negative, not positive. So instead of allowing positive particles to flow into the postsynaptic neuron, we actually get negative, right? Negative ions flowing into the postsynaptic neuron. And instead of resulting in an excitatory situation where the neuron can actually transmit that impulse, it actually has something that we call a hyperpolarizing effect. It takes us further away from threshold potential, further away from that threshold at which the neuron can actually fire. So it's almost like when you have a positive feedback loop and it moves you further and further and further and further away from homeostasis. Inhibitory transmitters move the cell further and further and further away from the threshold for which it can actually transmit a nervous system impulse, okay? So it inhibits the transmission of that nervous system impulse from one neuron to the next, okay? And this is because the outside of the neuron is much more positive than is normal. When we talk about just a bit of a graphic and which neurotransmitters do we actually need to know, acetylcholine is the excitatory neurotransmitter that is in our program of studies, okay? So that is one for sure that you need to know. Acetylcholine is an excitatory neurotransmitter, and this one is primarily for muscle function. So you'll notice it says it's at neuromuscular junctions. And remember, because this is excitatory, what this allows is it allows sodium channels to open and sodium to flow into the postsynaptic neuron so that the nervous system impulse can be transmitted. Now in this one here, you actually, instead of seeing a postsynaptic neuron, you actually see an effector here. And remember what the effector is. The effector is like the target of the nervous system impulse, right? So in this case, the effector is actually a muscle, right? So this is when the neurons have all communicated to each other, and then this neuron that we see here, the axon terminal of this neuron we see here, is the last neuron before we actually reach that muscle cell. So same thing, we have our action potential, right? Our electrical impulse moving down that acts towards that, that synapse. And then we have our vesicles, right? Here's our little synaptic vesicles that are little bubbles, our little carrier structures, little carrier organelles that can carry those neurotransmitters. So the little red kind of circles there represent, in this case, acetylcholine. And then you'll notice what happens as the action potential moves down. It forces the fusing of those synaptic vesicles with the membrane of that axon. And then exocytosis happens and the neurotransmitter gets put into the synaptic cleft and attaches to the little receptors here, right? There's those little receptors. And then of course, what ends up happening is that opens up those sodium channels and allows sodium to flow in to the muscle cell in this case. Acetylcholine, once it's done its job, okay, once it's facilitated the transmission of that impulse from neuron to neuron or from neuron to effector, as it is in this case, and it's not needed anymore, it's broken down very, very quickly by an enzyme called cholinesterase. And cholinesterase is also in our program of studies. And so what you really need to remember is that acetylcholine is an excitatory neurotransmitter, and then cholinesterase, which is present in our cells, will actually break down acetylcholine so that it doesn't just stay in that synaptic cleft. The other thing that can happen too, besides the breakdown with cholinesterase, is something called reuptake. And so sometimes when you have those neurotransmitters kind of in the synaptic cleft, they will be taken back up into the axon and repackaged into a vesicle, and that's called reuptake. So we can have the breakdown through the enzyme cholinesterase, or we can have reuptake and repackaging into a vesicle for future use. Acetylcholine, once it's removed, those channels obviously close and the neuron begins to prepare for another nervous system impulse. So when we talk about inhibitory neurotransmitters, we actually don't have any in our program of studies that we need to know. So, like specifically, so you need to know what an inhibitory neurotransmitter is, but we don't need to know any specific ones. Like GABA, for example, is a super common inhibitory one, but we don't need to know that. We need to know acetylcholine. We need to know acetylcholine is excitatory and it's broken down by cholinesterase, and we need to know norepinephrine. Okay, so norepinephrine is the other excitatory neurotransmitter that is in our program of studies and that we need to know. So norepinephrine is also an excitatory neurotransmitter, so it acts the same way in acetylcholine does, except remember that acetylcholine is more at neuromuscular junctions, responsible for alertness, learning, and memory as well. And norepinephrine, on the other hand, even though it's excitatory as well, it is responsible for kind of helping us maintain our fight or flight responses, okay? When it's given as a medication, norepinephrine is used to increase and maintain blood pressure in limited short-term health situations, okay? But it is an excitatory neurotransmitter, so it acts the same way as acetylcholine, just for different processes.